Good afternoon, everyone. Welcome to the first of the academic year Wednesday afternoon lectures to be given this afternoon by a distinguished professor from Hopkins, Andy Feinberg. Those of you who are new to NIH, I hope you will make a practice of coming here on Wednesday afternoons, because we line up at this particular hour in the middle of the week, really quite a remarkable number of speakers from all over the place at the top of their fields, oftentimes asking them to put forward not just stuff that we all know about, but provocative new ideas, and today will be no exception. The list of some 35 speakers that is lined up for this coming academic year is available on the web, and also there are various posters around that you might note and try to get those on your calendar. Welcome also to all those who are watching on the web, because while Missouri is nicely full here this afternoon, I know there are hundreds of you who are trying to multitask there in whatever location you're in the middle of, and welcome. Glad you could join us in this fashion. It's my great privilege as the NIH director to be able to introduce many of the Wednesday afternoon lecturers. When I have the chance to do so, it's always fun to remind myself a little bit what field they've been working in. In this instance, this is somebody I know quite well, because Andy Feinberg and I were faculty together at the University of Michigan back in the 1980s and early 90s, and have remained friends and colleagues ever since. He is, in fact, a remarkably creative scientist who has, over the course of several years, now a couple of decades, contributed in very significant ways to our understanding, particularly of epigenetics, a field which, perhaps when he started into it, was almost non-existent or considered to be a little squishy and Lamarckian, and now, of course, is the subject of a great deal of interest and a great deal of NIH-funded work and a great deal of insight, understanding about how modifications in terms of methylation of DNA, in terms of what happens with the proteins that bind to DNA, have a profound impact on biology and medicine. Andy started out as a geek in the mathematical arena. I think I remember that he was hung up on Fibonacci numbers for quite a while. Maybe he still is. Went on to get some undergraduate training at Yale, and then moved to the Hopkins program, which was accelerated, getting his MD in 1976 there at Hopkins. Went on to get an MPH there as well. And after a little bit of self-investigation about what his calling was, landed in Bert Vogelstein's lab at Hopkins, which he said was like going to heaven, because basically it was everything he was looking for in terms of scientific creativity, opportunity to explore new frontiers. And there it was that Andy and Bert defined for the first time hypomethylation as a signature of cancer, something that had not been previously noted, published in 1983. After that, he did head off to the University of Michigan, and his lab and my lab were in adjacent buildings on the same floor, so there was a lot of back and forth between our groups. I do recall, because he and I were also both trained as medical geneticists, that we would share experiences going to the clinic, sometimes in Ann Arbor, sometimes in outlying parts of Michigan. And maybe he could correct me, but I think it was there that he first encountered the Beckwith-Wiedemann syndrome, maybe in Petoskey even, which ultimately became a very significant insight for him and then for the rest of us about how epigenomics can play a role in medical illness in a way that had not previously been appreciated. After spending some 8 years at Michigan, he went back to Hopkins, where he's currently the Gilman Scholar, the King Fahd Professor of Medicine, Oncology, and Molecular Biology and Genetics, and the Director of the Center for Epigenetics. He's a member of the Institute of Medicine, elected to that role, and proud to say he is also a fairly recent successful applicant for a Pioneer Award, one of NIH's ways of trying to provide exceptionally creative investigators with the freedom to pursue ideas that might otherwise not fit the traditional R01 mechanism, and which he has used in a variety of interesting ways to look at a stochastic epigenetic model for evolution, development, and disease, studying, among other things, honeybees. So, I don't know exactly what Andy's going to put in front of you today, but I'm sure you're going to find it interesting and provocative, and please join me now in welcoming Dr. Andrew Feinberg.
So you didn't embarrass me. Well, I'm just thrilled to be here, and I'm just incredibly grateful to Francis for that exceptionally kind and generous introduction, and to Luigi Ferrucci for nominating me to give this lecture today. I wanted to point out that the Beckwith-Wiedemann patient was brought to my attention by Francis, who, among his many other unbelievable skills, is an incredibly gifted clinical geneticist, and recognized not only that this patient had Beckwith-Wiedemann syndrome, but it would fit my research interests really well, and it was transformative for me, actually, for several years. So just thought I'd share that. Anyway, so I'm going to talk today about the epigenetic basis of common human disease. Antoine, my microphone is good? It's sounding okay? Good, great, okay, thanks. So this is where I work, in the Center for Epigenetics. This is the Google Earth view. So like all Hopkins buildings looked at that way, it looks like a parking structure, but in fact there are laboratories ducked in there. And so all geneticists are interested in the basis of phenotypic variation. And, you know, we're actually interested in all of them, but of course, this is the National Institute of Health, so we're funded to look at the ones that are relevant to human disease. And I would argue that if you ask genetically what's the basis of phenotypic variation, and you ask, say, how is a human different than a chimpanzee, I would argue that, with all due respect to the great Dr. Goodall and to her friend, the differences are comparatively modest, at least when you compare to, say, something like a plant or a dictyostelium, which is what I used to work on before I came to Bert's lab. And that they're entirely explained by information we currently have. We don't know how to interpret it completely yet, but we have it all. We know what the complete sequence is of these different species, and that defines what those differences are. But I think an even more complicated question than that, as hard as that is to understand, is what's the difference between the brain and the heart and the liver and the colon? So here are all these different tissues. And in fact, it's pretty easy to see that the stomach, say, of a chimpanzee is far more different than the eyeball of a chimpanzee than the stomach of a chimpanzee is from the stomach of a human being. And yet, those tissues know what they are. They have information that defines their function and tells them what to do, and they remember what they are when those cells divide. And that's what we really mean by epigenetic information. So there are modifications of the genome that occur during development and define tissue-specific differences other than the DNA sequence itself, which is identical across the tissues. Now, it's even more complicated than that because, as you may know, Dr. Ferrucci is, also has an address in Florence, Italy, and I was over there not all that long ago, and I encountered this 8-foot guy named David, and I invited him. He's sort of the perfect example of human development, to visit us in Baltimore, and he went down the street and had one of these double Whopper cheeseburger things, and I'm sad to say he sort of wound up like this. So, I mean, the point is that our environment shapes in a remarkable way our phenotype, but not through the genes themselves, but through influencing how the epigenome might take place. Now, the person who coined the term epigenetics was Conrad Waddington, who wrote a series of monographs, 3 of them, in the 1950s. He was at Cambridge University. And he said that epigenetics means phenotype that arises from genotype through programmed change and interaction with the environment. But he looked at it as an entirely programmed process. It might deviate, but it's going to be pulled into a particular pattern of, say, a tissue development. And his original monograph was something like this, where you have a pluripotent cell, and it can become a liver or a spleen or a colon or whatever. But then people pointed out that water runs in the other direction, so, literally. So his friend Piper, the landscape artist, redrew a new picture, which is the one that people see, of a ball rolling down the hill, and here's the pluripotent cell, and although the environment might push things up in this way, they eventually roll down into this pathway or this one, which are each of the different tissue types, and that's controlled, according to Waddington, entirely by your sequence. And he called that process canalization, actually because of this cartoon. The modern definition of epigenetics is a little bit different and a little bit more flexible and a little more information-based, and that is modifications of DNA or associated factors that have information content and are maintained during cell division other than the sequence. I alluded to that a little bit earlier. And just to give you an idea, here's a cell, here's a gene that's making an RNA active, and here's a gene that's transcriptionally silent. And there are a number of things that happen. One is DNA methylation. So that can occur at C followed by G. People call that CPG. I don't know why, because of course there's a phosphate between the C and the G, and they don't say tippa-tippa-ta box. But nevertheless, it's CPGs. But we also know that there's non-CPG methylation, but we know of no mechanism to copy that, so it isn't purely epigenetic information. This is copied. I don't have time to go into all the mechanisms, but there's an enzyme that does that, recognizes it, copies it. And that's associated in general with gene silencing, and it was thought, until some recent studies that I'm going to talk about, to be enriched or almost entirely at dense regions where there are many CPGs that have been called CPG islands. Then, of course, there are histone modifications. You've got some great histone biologists right here at NIH, like Shiv Grewal and so forth. And no offense to the 30 others who must be here too. I just am friends with them. There are some of these post-translational modifications of histones. So you see the DNA is wound around those histones that are associated with activation, and other ones associated with silencing. And they're quite different. It's either this modification is for activation or it's for silencing, but never for both. And then there are proteins that recognize those complexes, and I've listed some of this here. And then those are trithorax for the activation, polycomb group for the silencing. And then, interestingly, there's this issue of chromatin structure, which I've become very interested in. You're going to hear more about that. And that has to do with both the density of the nucleosomes and how the DNA is coiled within the nucleus. And it's not put into the nucleus the way that my daughter packs her suitcase when she's going somewhere, which is very interesting and no order to it, but in an ordered way. Now, I'm going to talk about some puzzles that were bothering me about 4 or 5 years ago, really haunting me. So one is this issue of the missing heritability of common disease. It's a bit of a sensationalized term, except that people in the field use it themselves in their own papers. And it means that common variants, at least, can explain, depending on the disease, 1 to 20% of the genetic variants of a disease. And it's a little bit puzzling. Where's the rest of it? And there are many good explanations, including rare variants, copy number variants, things that we can't just identify because of the way that our studies or the epidemiologic cohorts were designed in the first place, and so forth. It's a very active area of study, but still it's something people wonder a lot about. And then there was this very provocative paper in Science Translational Medicine from my former mentor, Bert Vogelstein, and he didn't mean any harm by the paper, but it was a twin study, and it showed that it's difficult to explain the contribution to disease greater than a factor of about 20%, and there must be a very strong, therefore, environmental component to disease. In a way, it's a tautology, but you cannot imagine the storm of criticism that it gendered, and there's very humorous give and take in the, all well-meaning really, in the comments and replies that appeared after that paper came out. But I wondered, okay, it's one of these things that was puzzling me, could epigenetics play a role? Now, as Francis alluded to, many people who think about epigenetics, certainly in some of the things that in the popular science, not so much anymore in the scientific community itself, but still there's still a component, that there might be a Lamarckian role to epigenetics. And that idea is that, and you have to read Lamarck himself to see what he's really saying. It really is loopy the way at least he describes it, but there are these fluxes, and they're caused by environmental exposure. So the giraffe occurred because a horse had to stretch his neck to eat a leaf, and then the fluxes got in them and got transmitted to the progeny, and then they also had a longer neck because of those fluxes, and that carried on for further generations and so forth. And there's the little picture of Lamarck on top of this, and I'm sort of ridiculing the idea a little bit. I'm not saying that there isn't some component of transgenerational inheritance. I think that's been well demonstrated, in fact, that the environment can have an effect, say, prenatally, as was shown in a recent important paper from Ann Ferguson-Smith that was in Science, but that it would be stably transmitted, and here's the important thing, over and over and over again, despite germline reprogramming, and undergo evolutionary selection is difficult to understand, at least for me. Now, I'll come back to that in just a moment. So about 12, 13 years ago, I became very interested in whether or not one could start to explore the epigenetic basis of common disease generally, including cancer, but other diseases as well. I mean, cancer is the one where people had finally come to accept an epigenetic component, but what about all these other diseases, particularly where environmental exposure might be important? And in 2004, a student, Hans Bjornson, and Danny Fallon, who's an epidemiologist I work closely with, and I wrote this paper in TIGS suggesting that we didn't call it epigenetic epidemiology. We had a much worse name for it. That term really belongs to Stefan Beck. But our suggestion was that you could integrate environmental exposure, DNA variation in modifying the epigenome, and that it might even lead to quantitative phenotypes. So in other words, it would integrate genetic and environmental exposure into the epigenome to define phenotypes. That doesn't mean that explains all of genetics or even most of it, but it might explain some of it, particularly that which is related to the environment. That was the idea. An extra thing that we sort of said in here, but it's, I think, a neat point, is that you could get quantitative traits over modifying the epigenome without necessarily having to invoke lots of individual variants. In other words, they could either integrate individual variants or environment together with genetic variation to give quantitative traits. So we put in a grant that I was thrilled to get funded for a Center for Epigenetics at Johns Hopkins through the Genome Institute to look for new tools. That's a program actually was created by Francis when he was the director there, to develop new tools and apply them across the whole epigenome and also to populations of patients. The first thing we did, with the help of Rafael Irizarry, a brilliant statistician, is we developed this array-based method which we call CHARM. It's an ill-fitting acronym, but it really is meant to appeal to the moniker for Baltimore, which is called Charm City. It's actually really nice, okay, Baltimore. I know people snicker because of TV shows and all that, but it's a great town. That enabled us to interrogate the genome at a very much greater scale than people had done before. The typical thing was looking at something like 14,000 and then 40,000 of these islands, but this is a very different scale, up to 4 million sites. And then we didn't invent whole genome bisulfite sequencing, but there were some very clever developments from people like Ben Langmead, who invented Bowtie, and Caspar Daniel Hansen, who's a brilliant statistician, now completely independent at Johns Hopkins, at developing tools to reduce the cost of analysis, very clever ways to improve throughput for extracting epigenetic information from sequencing. And the latest bee in my bonnet, which we're just getting started at doing, there's a big center at Hopkins for keeping track of all the stars in the sky. And Alex Ali leads it, and the idea is to try to use the graphical processors in massive parallel ways, these cheap little processors for $50, and just swap them out and so forth. And so we're hoping to use that power, because I'll tell you, the biggest bottleneck in our research, and I hear this from other investigators too, there's probably a paper on this, is the computation. Now, it's not the experiments anymore, it's just the computer time and the analysis. So I'm going to show you the second big puzzle that was sort of eating at me that came from a study. It was really our first big study of the epigenome of both normal and cancer cells. And this is the result of that CHARM analysis. So we're looking at millions of regions of sites of DNA methylation across the genome. And the question we've asked here is, how is colon cancer different than the normal colon mucosa that came from the same patients? Which is actually the original question I asked with Bert Vogelstein decades ago. But now we're using CHARM to look at DNA methylation at all of this. And this is a heat map. Red is more methylated and blue is less methylated. And these are the 500 sites that most distinguish in a supervised cluster analysis the cancers from the normal. And you see they're really very different, fine. But then here's a great big surprise. If you look at the dendrogram over here, so these are the sites, the 500 sites of different methylation, and you just put them over here and do an unsupervised clustering and ask from autopsy specimens, how well do they discriminate the normal spleen from the liver from the brain? There's no colon in there. They do it completely. And a way of saying that formally is that the cancer differential methylated regions, or CDMRs, are the same thing as the tissue differential methylated regions. That's a big surprise, because it means the very things that are telling a colon cancer that it isn't normal is the same thing that, to go back to my original analogy, is telling the eyeball that it isn't a stomach. And why should it be general like that? And how's that really related to function? So it was a puzzle. The other thing that I noticed is that Yeah, so the changes are the same. But the epigenetic targets are hypervariable. So it's not that surprising that there's this great deal of variability. You could see it in the pattern of the tumors compared to the normal, but there even is in the normal itself. And look at this. There is clearly a great deal of variability in the normal tissue as well. Now, that could be caused by the genome sequence, right? Because sequences could control DNA methylation. But if you look at inbred mice, so that's like these samples right here and those samples right there. Look at the variability. These are brother-sister matings, inbred strain. They still show variability, even though those regions in the genome are conserved very closely from human to mouse. So they're highly conserved, but variable regions from animal to animal, but the locations are conserved from one species to another. That's really strange result. And it was sort of eating at me, as well as this issue about missing heritability. Not so much that, but whether epigenetics could fit in or not, because it's hard to make the argument if you can't. I don't think you can rely on Lamarckianism. So this thing happened to me. Okay, so I call it my epiphany. And it's sort of a joke to use the word, but it's like a way of saying a good idea at a church. So I was just touring, sightseeing with my son in London. And what really happened is we wanted to go up Big Ben, and there's a sign out front that says in very polite English, if you're not British, get lost. Okay, so that's not the words they use. And right next door is Westminster Abbey, and they had a sign out front that said, it's the 150th anniversary of Origin of Species, and Darwin's here. He's dead, but it's still fun to see him, you know. Again, I think I've changed exactly what the language was on the poster. So we waited and we went in, and I found myself standing on Darwin's grave, okay, and that took a surreptitious picture. Right next to Darwin's grave is Newton's grave. Now, what I'm not showing you is that there's this velvet rope right here, and that's because the British do not want you to put your filthy feet on Isaac Newton's grave, but they don't have any such misgivings about Darwin. And they also decorate Newton's grave with the cherubs and things like that. They really like him a lot. And just above Darwin's grave is a plaque to Dirac. So Dirac was one of the people who founded quantum theory. And I thought maybe he's buried standing up. The laboratory, when I got home and told them this whole story, said, no, it's an urn. He's there in an urn. And I was explaining this to some people in Florida at the University of Miami, and they said, what are you talking about? He's buried in Bonita Springs, Florida. So it's just a commemorative plaque. But I saw this, and I thought, well, but there's nothing like that here in biology. And so maybe there's something about this stochasticity that's the core element of quantum theory that would apply to biology and maybe solve this conundrum about where epigenetics fits in and where that variability is coming from, and maybe that would make sense in evolution. So this is the idea I had, so that you could have stochastic epigenetic variation as a driving force of development and evolutionary adaptation. And the elements of the model are, first of all, you don't need this in single-cell organisms, and in fact there are mutational mechanisms that do just what I'm saying in bacteria. But for a complex species and multicellular organism, I thought it might make some sense. The idea is that stochastic variation plays a fundamental role during normal development, like quantum mechanics does in physics. Now, bear in mind, these are not quantum mechanical distances, but the idea is, just like Niels Bohr, you know, if he was brilliant and everything, but if they're actually completely correct that an electron is only in this one little place, and for a chemical reaction to take place, it has to go into that one particular place over here so it can be shared between these two atoms, it would never happen. The only way it ever happens is if there's some sort of a probability field and there's some interaction. Similarly, I think it makes more sense in developmental biology if the ligands and receptors that are responsible for example, renal development, which I know something about from having studied Beckwith-Wiedemann syndrome, if there's a gradient or a variability in the expression of both, so that some cells, like from the signaling molecules that are coming, say, from the ureteric bud, would interact with some receptors that are there at the right concentration and location in the condensing mesenchyme to lead to the glomerulus, for example. And maybe that's the reason why so many cells die during development. You need stochasticity so that at least some things take place. That was the thought that I had. But that this stochastic variation could be mediated epigenetically. And if that's the case, then the degree of stochastic variation could be controlled by genetic sequence. So there's no problem with Darwin. This is not Lamarckian. You would have genetic sequences that lead inherently, by the nature of their sequence, to epigenetic variability in the genome. And why would that make sense? Because genetic variants that increase epigenetic plasticity could increase fitness over evolution for traits that are affected by an environment that fluctuates. So the environment's one way, and then it's polarly opposite in an unpredictable way, and it goes back and forth from time to time. After periods at which selection would take place, you would select for such variants. It's a little bit of a complicated idea, and I'm going to show a little movie to illustrate it. So here's the conventional theory. Let's just say that you have 100 people. Now, I'm going to be talking about like sizes of people and dying and things, but I'm not really talking about people. I'm talking about little circles that I'm calling people. So, you know, if I'm saying it a little lighthearted, these are not actual people, okay? But let's say that you have 100 people, and they have varying sizes denoted by the size of the circle. So some small, some big. And that's controlled genetically, so that the red has 3 genes for being big, and the blue has 3 genes for being small. So under the normal way we think about evolutionary biology, the large survive, say if there's abundant nutrition and resources, because you'll get a bigger guy and he'll win the jousting competition and we'll get the girl and all of that. And the next generation will have a larger population of big people. But life on the planet Earth is not fair. So you might select for being big, but then nutrients might disappear for some epoch, you know, some number of generations. Then you'll select for being small, and then you'll be selecting for being big and small. So let's play out the movie and see what actually happens. So what would happen is you would be selecting for big, big, big. They're getting bigger, 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 and now you're going to select for small. Too bad everybody dies here, and now they're little, and now they get big again. And, you know, you get these huge die-offs in the population. When we proposed the model, we didn't know, because this came out later from the 1,000 genomes, but there haven't been these large die-offs in the human population over development. The selections happen in situ within existing populations. So what about the model that I'm suggesting? So now, in addition for selecting for the size, you're also selecting for the variability itself. So it's just the variance, really. So you have genetic sites that are selected for variance, not for mean. So, well, initially it's going to be exactly like it was before. Then you're going to have this huge change. But after enough fluctuations back and forth like this, eventually look what happens to the variance genetic selection. It takes off. And you wind up with a population that's extremely heterogeneous, that includes some people who were big with genes for being small, and some people who were little with genes for being big. And I think that's what our population, in fact, is like. And I think that's what might make it a little bit more difficult to understand from purely genetic data what's happening in terms of disease phenotype. So how do you test this idea? Well, the prediction of the idea is that there would be a thing that we would call a variably methylated region. So in an isogenic individual, it would be prone to a high degree of variability. And they, in fact, exist. This is a charm plot. That's more methylation, less methylation. And here's a region that shows a great deal of variation in inbred mice in the spleen. And here's another region that's just like that as well. So they do exist. And if you want to know if they're important in development, you do one of these Go annotation analyses for ontogeny, and you find out that they're involved in anterior-posterior pattern formation, organ morphogenesis, development of the central nervous system, development of the gut, development of the mesenchyme. I've never seen a chart like that where there are so many important processes that are all showing up for the same phenomenon. So how might this be relevant to disease? Well, the first disease that it seemed to possibly make sense is cancer, because cancer involves repeated changes in the microenvironment. So in a way, a cancer cell selected for first surviving like as a colon, but then being selected for being able to live in that space as it's invading, and then selecting for being able to travel through the blood, and then maybe landing in the lung, and maybe another cell lands in the liver, and then it remetastasizes somewhere else. So in fact, they're subjected to huge varying environmental changes, and like oxygen being one of the principal ones, right? So that is first very hypoxic, and then new blood vessels grow in, it becomes hyperoxic, and so forth. So we did a study to find out whether or not the sequences of change in cancer are changing more by mean or more by changing by their variance. and at the level of DNA methylation. So this is what's called a principal component plot. Basically, it just indicates by how close you are, how similar you are to other samples, but it's measuring across the whole genome. And you can see that normal tissues cluster very closely together: the breast, the colon, the lung, the thyroid, Wilms tumor, our friend from Michigan. If epigenetics was like mutation, where you have a defined change that goes in a very precise way from normal to cancer, you would go, say, from the normal lung to a new pattern of lung cancer. But that's never what you see. What you see is this. You get this huge stochastic variability in the methylation pattern at the very sites that define normal tissues. It's like a formal proof of what I was showing earlier visually in that first heat map that I showed at the very beginning of the lecture. And with as few as 25 CpGs, using this hypervariability measure, you can distinguish cancer from normal universally. And when we continued this work and then took it to the level of whole genome bisulfite sequencing, we got a new insight into what I discovered with Bert back in 1982 in this Nature paper. And by the way, so there's a picture of us back then. And here's a really interesting thing for the young people in the audience. When I look at myself in the mirror in the morning, that's what I think I see. Bert tells me the same thing about himself. Okay, it's really interesting. Anyway, so this is what we saw from whole genome bisulfite sequencing. There are these gigantic regions. So let me give you a picture again. This is from sequencing now, not from arrays. This is 1 million bases of DNA. That's highly methylated and that's low methylation. This is normal colon is blue and the colon cancer is red. And there is not only this reduced methylation, which was the original observation, but it's hypervariable too. And not only is it hypervariable, but it's associated with huge variability in gene expression. So these are individual genes within these regions. There's the normal, and here's the variability. Even when the genes are getting turned off, which happens sometimes, they're hypervariably turned off as well. And most of the genes that we know are involved in invasion and metastasis lie within these giant regions. It's a half of the genome is in these big blocks, and it represents 1/3 of single copy genes. This is a lot of stuff in the genome that seems to be in them. These are really giant VMRs from the nomenclature that I gave earlier. So what are these big blocks? Well, they turn out to be something else that we had been studying a little bit earlier. In this Nature Genetics paper from Bo Wen in the lab, we described these large organized chromatin lysine modification regions. So in other words, these would be things like H3K9 dimethylation, H3K9 trimethylation, H3K27 trimethylation, that we noticed originally just in normal tissue. This is a chromatin plot now. These are 20 kb regions, but these could be up to megabase in size that are just normally have this increased chromatin methylation associated with heterochromatin. And they correspond with a region that Bas van Steensel had identified associated with a nuclear lamina called lamina-associated domain. So they're highly corresponding, and that's what those blocks in cancer represent as well. Importantly, we showed, and others have shown as well, for example, Kim et al. In this Genes in Development paper, they're not present de novo. They're things that develop during differentiation, and there are even tissue-specific differences within them. Now, I had a postdoc in the lab, and this is an accidental discovery. Oliver McDonald, he's a pathologist, and he did an experiment I suggested where he looked to see whether an injury response, basically EMT, epithelial mesenchymal transition, is induced by TGF-beta, involves changes in DNA methylation. There were a couple hints in the literature that it might, and there was no evidence of this at all, at least in the acute stage. But Oliver, of course, stains everything with hematoxylin and eosin, being a pathologist. And he said, Andy, by the way, look how pale those nuclei are on exposure to TGF-beta, just by the eosin staining. No, the hematoxylin. I forget, Francis, which is it? Hematoxylin. I knew he would know that because he knows everything. Okay. And you could see that the TGF-beta is reduces the H3K9 dimethylation globally. And moreover, you can see the locks disappear. So there they are, visualized by the LADs, okay, which are the same thing, and they disappear on this exposure and they come back when you take this off. So it's something that's normally reprogrammable in response to some challenge like TGF-beta. So I suggested then that there is a dynamic landscape. It's different than what Waddington said. So it's not fixed and it's not totally gene determined, but it's adaptable. So these valleys can become much flatter and allow transitions from one cell to another, and that's itself induced by chromatin structure within these domains, both changes in the interaction with the lamin, but based on things that other people have done, and that I pointed out in this review paper, in cell chromosome-chromosome interactions as well. And so there's this deformable plasticity that's important in normal development that gets back at what I was saying earlier, that you need to have some mechanism in normal development and also in response to injury and other challenges for adapting your program. But you could sometimes do this by altering epigenetic plasticity and increasing stochasticity in defined regions. So this is just a model. I just want to refer you to this thing from there's a subsequent paper in Nature Reviews Cancer with a little bit of math to go with it, but it's not that hard to model how this might occur, flattening the valley and doing things like I'm not going to go into all the names. There are more names for the mathematical formulas than there are even in biology. But the essential idea is that even if you had, say, a deterministic element pulling things back to a normal level of, say, methylation, say this is like a Hookean spring kind of very simple process, you could add to it a variable stochastic factor, and that if that was relaxed, you would get this variability. And if you model that, the data that you get in terms of distribution of DNA methylation between normal, adenoma, and cancer matches very much the reality of what you see. This has been published, so I'll just refer to it briefly. This is data that I think are published. I saw a galley a couple days ago in Molecular Medicine, and this is also from Winston Timp working with Rafa and me. And I want to show you this. It's kind of amazing. So we saw, this change in DNA methylation in these block regions, as I mentioned, this large-scale hypomethylation. But what this result shows, and this is just an example of breast cancer, but here is the region of islands, all normalized over a central region, and then the nearby regions that we call shores. We've defined these hypomethylated shores in cancer and these hypermethylated islands. Look how in the cancer, there's this, if you compare, say, the breast cancer outside these block regions, there's high methylation, and you go to the island and it's low, and then it goes back up normally. Inside the blocks, these are islands inside the blocks, you get this hypermethylation of the islands and hypomethylation of the shores. But nothing much happens outside the blocks. So even the changes locally that people have spent the last 20 years looking at around CPG islands, both in them and nearby, seem to be largely an attribute of these large domains of variable methylation that seem to be able to be controlled by these chromosomal mechanisms. In other words, sort of the Pac-Man result. The blocks kind of are eating the islands and the shores, at least mathematically. There's some data that I'll show you that's unpublished where we've been looking at aging. And this is data from Amy Vandiver, also working with colleagues at Hopkins from the skin, from the dermatology area, Luis Garza, Sewon Kong. These are these hypomethylated blocks that I just described a little bit ago, and that's just a quantitative depiction of them. We see them in non-malignant tissue that's been in photo-aged older people. Not really without the combination of both of those things. So we think that the environment induces these changes over time. There's work, this is like my most favorite paper to show you because it isn't from me, but it sort of supports some of these ideas. This is work that was done by Andrew Teschendorf and published in Genome Medicine in 2012, where he showed that if you take biopsy specimens from women who either later went on to develop cervical cancer or didn't, why would you have that? Because it's a real conundrum, even with the vaccines and everything, following people making histologic diagnoses. So a lot of women get these biopsies, and then later on, most of them don't have cancer when you do the biopsy, but later on some of them develop cancer. Using the variability testing, you could tell who's going to develop cancer later, where you couldn't do that with the testing of mean differences in DNA methylation. So I think that this variability occurs early, and it's predictive of cancer development. We don't know mechanistically why exactly, but we have an idea about that. The idea is this. So you've all heard about the hallmarks of cancer, and I'm not questioning their existence, but I'm suggesting that what really might be happening is that the center of all of this is the epigenome. And the epigenome needs to be stable for a given tissue. And if something derails that stability and leads to increased stochasticity, it will allow natural selection of those cells at the expense of the host. And that model will only matter and take place if there's repeated changes to the microenvironment. And of course, cancer arises from repeated cycles of injury and repair. Many types of cancer are known specifically to occur because of that, like cirrhosis of the liver, even colon tumors as well. I mean, we know that there are primary epigenetic defects that can lead to changes in the epigenetic machinery, but I think the most compelling evidence for this idea is that most of the mutations that have been identified in cancer are affecting the epigenome, and I would argue that they're affecting epigenome stability specifically. So it's an evolutionary process very much like the one I was describing earlier. Now let me come back to talk about some other diseases in the final 10 minutes or so of the lecture. So I want to talk about this idea of combining epigenome-wide association studies with GWAS to look at common disease in a practical way. So the first effort that we took in that regard outside of cancer was an autoimmune disease, rheumatoid arthritis, which is common in the population, about 1%. It's about twofold more common in women. This is work that was done by Yuan Liu with colleagues at Sweden, including Thomas Ekström. And the end result of that was we identified a region that changes its DNA methylation under the control of SNPs in this region that is nearby the HLA cluster, where there are defined changes in amino acid sequence caused by sequence variation, and we think it has something to do with the expression of these HLA family genes. This was a validation of these results by independent methods on independent flow-sorted samples showing these methylation differences are consistent across separate individuals. But here's what we really saw. So now here is a density plot of the methylation for a genotype that does not lead to rheumatoid arthritis, and here is for the homozygote genotype that does lead to rheumatoid arthritis. And then here's for the heterozygotes. Well, it's increasing the variability of methylation in that region. And these individuals are well, actually, and these individuals are sick. So if you only did the GWAS, you won't see this particular site. You do see the sequence encoding ones, but you don't quite see this. It's a little bit below the radar screen. When you combine it with this methylation assay, then you can pick it up. This is a very recent study, also done by Yuan, but with the help of the BLSA, led by Luigi, to look at what is the formal relationship across the genome between these SNPs that might be regulating DNA methylation. And this is the association of one SNP with another. That's what LD is, okay? And that's the distances for LD over about 20, 30, 40 kb. The methylation association is very small distances. But what's surprising about that is you can have SNPs that are down here that are regulating clusters of CGs that are located far apart. And here's the formal proof of that shown. These are from several different regions spread out over 60 kb, and yet they follow in the population a very similar pattern under the control of these SNPs. Now, why would that be? It's because the genome's folded. These are regions that are probably associated with each other in the not my daughter's suitcase packing method, but in the way the cell really organizes things along the nuclear membrane. And they don't necessarily lie within the same LD block. So that could, so that means that the integration of epigenetic information with genetic sequence could reveal patterns to human disease that we wouldn't know otherwise. And here's a supplementary figure from that same paper that show exactly how that might be. So here are several SNPs, for example, that are associated with metabolic phenotypes, but what they control in methylation is a little region. We call it a gene, just because we like to come up with really bad names for things. But here it is, and it's right in a shared promoter region for two genes that are very important in metabolism. So I think that might help to explain some of genetics. So let's get back to what else causes disease in the last couple of minutes. I want to show you very briefly a story that we're just about to send into a journal. Well, the environment, right? I mean, that is really the point, and it's something we have to address and figure out how to deal with. So like smoking, the sun, just getting old. That's a 100-year-old woman who looks exactly like my mother. And when I pointed this out to her, she didn't talk to me for about a week. Hamburgers, stuff that inflames the colon, like drinks and all that. By the way, whenever I see these kind of pictures in a lecture, I want to have a hamburger and a cocktail, but oh well. It's very difficult to study the environment and people. And epidemiologic studies are very expensive. They're heroic to do them, but they're very tough to do. The real issue is you can't control the environment in human populations. It's just not there'd be no way. It's like ridiculous to even think about doing it. So how else are you going to really study that? So we have turned to mouse, and a lot of my lab is now interested in figuring out these using new models of the mouse. So here's the data I want to show you. So we took mice, black 6 mice, and we either gave them a normal chow diet or a high-fat diet. And this is work of a graduate student, Michael Moltop, a terrific statistician, Andrew Jaffe, and the wonderful biochemical work of Will Wong in his laboratory with the help of Jolene Zarath and colleagues at the Karolinska Institute. And we take these different tissues from them, and then we did charm analysis, which is a pretty inexpensive way for getting a lot of data from these animals. And we asked, what does the high-fat diet do versus the low-fat diet in terms of DNA methylation? Also in terms of diabetes-like phenotypes, like insulin tolerance. And we do our typical thing, and we find lots of these DMRs that are related to either high-fat or low-fat diet or one of these phenotypes. We found 625 of them with genome-wide significance in the population. Then we said, all right, of these 625, 491 map onto our charm arrays in humans. And of those, how many are conserved? About 80% of those regions were conserved from mouse to human, which is really astonishing because none of them was within an exon. And then we found methylation changes using tissues that we got from our Swedish colleagues from either obese versus lean individuals or from bariatric surgery, from people who were obese and then they were treated by gastric bypass. And we found that 249 of them showed methylation changes and 170 of them were in exactly the same direction, even though this is 50 million years apart, the mouse and the humans. And then we mapped them to the diagram study for the diabetes risk. And bear in mind, I mean, this is largely pancreatic, this is largely adipose. So it's almost amazing that we would find any overlap at all. But 30 of them overlapped LD blocks and were significantly overlapping those regions over what the null would have been by permutation analysis. And so this is just a dramatic example of this. So here's the mouse data. Here's the exact same region where the obese individual has the same difference, and it goes halfway back after bariatric surgery. So just to show you that. This is the result. And what I've shown here is an ingenuity plot where I've shown our 30 genes and then just a couple that are pale just to show connections between them. And the first thing I want to point out is that 2 of these that we found using nothing to do with genetic analysis turned out to be top GWAS hits. So we have to be onto something that's meaningful for that to be right. And those are shown in blue, these 2 genes right here. But then we found that there were the other 28 or so were overlapped GWAS hits, but that had not risen to genome-wide significance by GWAS. But they have to mean something because they're so close in these pathways, like here to here, these to here, and so forth. And the other ones that are shown in purple are just conserved mouse-human DMRs that are showing changes in the same direction. So the plot itself suggests that there's something real about this mechanism, about this process, and the ability to go from mouse to human that might tell us something that we didn't know about diabetes or what diabesity, in other words, obese-associated diabetes phenotypes. But to sort of give the most convincing argument, we took 6 of these sequences that are shown with the red circles, for which there was not a single paper in the literature suggesting any connection to fat metabolism, diabetes, or obesity. And we did manipulations in cultured cells. Here's the data from 2 of them. Here's a control of overexpression. 5 of the 6 showed when they were overexpressed impaired glucose utilization, showing that they really are involved in metabolism in this way. So I think that this might be a way to uncover new targets that are, if not the primary generators of these phenotypes, they're certainly mediators of them, and they might be druggable. We might be able to come up with compounds that might modify the phenotype. So in summary, I think that epigenetics does tie environment to genetics and disease. That doesn't mean it explains everything. And I have to ask you, here's my usual disclaimer. I'm sorry I forgot to say it until now. Please do not confuse my enthusiasm for the ideas with certainty that they're right, because I don't possess such certainty. I just get excited about this stuff. Epigenetics clearly drives not just cancer, but it has a role in common disease generally. I think that's emerging to be a true story. This new stochasticity idea could provide a mechanistic foundation, namely nuclear architecture. And I didn't mention, but in Oliver's study, we were able to reverse those changes and reverse EMT by LSD1 inhibitors in that case. But we think that some of the drugs that are currently being used for cancer therapy might also have an effect on this as well. And they wouldn't come up in the usual, cell toxicity assays, but they still might be beneficial in this way. And this new field of epigenetic epidemiology could offer faster, cheaper transportation on the road to individualized medicine. But I think in combined with animal studies, which I think are a plausible way to go if you can bring these together. And I'm just very surprised myself at the conservation of these environmental epigenetic signals from mouse to human. What it really means is that these are regulatory pathways. You know, we're not selected to have disease, right? But these are regulatory pathways that are corrupted by the things that we eat or just by our longevity or exposures or whatever. But they're very important functionally, and they've been around for a very long time, at least for 50 million years. But thank you very much. Appreciate it.
Thank you for a wonderful romp through a lot of ideas, and it's time now for people to pose questions. There are microphones in the aisles, and ask you to use those so that people who are watching on the web also have a chance to hear the question. So please don't be shy while they're thinking about it. Andy, in terms of what you're seeing in terms of this variability of methylation, which is quite striking when you look even at inbred mouse strains, so it's hard to blame that on DNA sequence. What happens if you look, assuming you can, at single cells within a given animal? Is there as much variability between cells as between animals?
We would, oh, okay. So we don't know, because this is a technology that's just coming along. We're working hard at it. A lot of other labs are working hard at it. But we think it probably would be, at least for cancer, because we think we see this variability in cancers at the earliest stages at adenomas. And I remember even back when I was doing work with Rick Boland looking for the conventional genetic changes, it's in that very early crucible of early stage malignancy that you see this emergence of lots of variability within these lesions. And then you get selection and then almost a homogenization over time. There's a lot less variability in these advanced lesions there is than even in the early primaries. But our data suggests that something like that would be going on, at least in tumors. But the really interesting question is how much is this variability present from cell to cell? And I think that's something we have to answer. It's a great question.
Thanks, Andy, for this. Very interesting. So, you know, some of the liquid malignancies, like AML, they show normal karyotype. They show nothing on fish. So they're very normal genotypes, very normal sequence. Mm-hmm. Could your stochastic epigenetic theory apply to that? Play a role in development of these?
It might. I mean, we've actually looked pretty hard at that. We've been doing some experiments on AML, and what we're looking to see is whether or not there are defining signatures for the really dangerous phenotypes of engraftment that are associated with poor survival. And we think so, in fact. But this explosion of a high degree of stochasticity and so forth, we actually find very little evidence for that in AML. If it's there, it might be confounded by the large number of mutations that are present in the patients who have the disease. But actually, as much as I'd like to say that liquid tumors are like solid tumors, our own data don't really support that so far. Yeah.
Over here. Yes, you spoke about pre-neoplastic changes epigenetically prior to morphologic changes in cervical cancer. Could you elaborate on the type of study you used, the type of sample? How did you do that?
I can't, because it's not my study. That's why I liked it so much. So no, I don't mean to make light of your question, but I don't know, right off the top of my head to remember the details of the study. I don't remember that well. But it was Andrew Teschendorf, and there were two papers. One was on cervical cancer, and one was on breast cancer. They were both in genome medicine or genome biology. And what they did, he moved to Singapore, but he was the statistician of record for the women's gynecological cancer registry. So they had all of these samples, and they'd been doing originally 27K arrays. I do remember that. I know he's moved more recently into the 450K Illumina array. And then what he did is really amazing. Actually, the reason I really love the paper is because, I mean, he read our first paper on the idea from the, Westminster Abbey and the suggestion that cancer might be involved. And he actually cites that paper, which he didn't have to do, that was really generous of him, and said that, he's going to develop a statistical tool for looking at this variability. And both in our Nature Genetics paper and in his paper, we had to borrow tools from econometrics, because in biology they really don't use this sort of a mathematical analysis. So we used something called the, I'm going to regret it for the rest of my life, I'm blocking on the name. I think it's the Levine test. I did remember it. And he used his own test he invented called Evora. I don't know what it stands for. And they're both measures of something called heteroskedasticity, which is something that the Wall Street guys use to make all that money that we don't get.
Andy, I asked you this question 2 years ago, but I'm going to insist.
Okay.
So stay on the variability of methylation. If you compare a young individual and an old individual, what do you think is going to happen? Do you find increased variability or not?
Yeah, so, well, we don't know, I mean, that's the great experiment. Here's why we don't know. We don't know because there's this issue about cell type. So your cell type changes as you get older. And the samples that we've been examining so far are from blood. Even if you take blood mononuclear cells, there's still several different cell types. So until someone can do a study, which I think is a fantastic thing to do, and I know you're doing it, to isolate purified cell populations from individuals the same individuals actually over time, you can't really answer the question. So I think it'll change. My guess is that you would have, I'm just guessing, you would have some reduced plasticity, that might account for the reduced adaptability of aging to stress. I mean, a lot of aging problems are related to the inability to respond to a rapidly changing environment quickly and then go back. So that would be what I would guess, but that's just a guess. It's not based on facts.
So I was wondering if you could elaborate a little bit more on this idea of missing heritability, comparing the role of epigenetics and of interactions between genes, epistasis, which is also something that is missed by a GWAS study.
Yeah, so, well, I don't know that epistasis is really missed by GWAS studies. I mean, I think some fantastic studies. Yeah, the power is limited. If you could do a GWAS study where you could control the environment, right, on your population, then I think, I mean, that's the problem. I think that it's not that there's a flaw in GWAS. It's just very hard, really, to look at an outbred population, which is what we are and hope to remain, and with many different exposures, which also, given the there are both of our interests in gastronomy, Francis. I mean, we also hope that there will be a great deal of diversity in diet as well that will persist. So it's going to be difficult to get at particularly the phenotypes that are associated with environmental perturbation itself. It's going to be difficult to power that. And I think that's why mouse can help. I think I wouldn't use the term missing heritability if the GWAS people themselves didn't use the term, because it's a code word to tie in with what they mean. It's not so much that it's missing. It's just different things you have to do to resolve out different questions. What I'm really arguing in our diabetes result is that some of these things are going to be purely epigenetic that we saw. They're probably downstream of the phenotype changing, which doesn't mean that intervention still wouldn't be beneficial medically, because there's going to be feedback loops that they'll be important in. And certainly the genetic manipulation experiment suggests that exactly that, right? You can manipulate the gene, you're going to get a change in glucose uptake. So even if it's downstream, it's important. There are going to be things that are intermediate and mixed, and there are going to be things that are strongly genetic, but you need to look at all of it. I mean, even in code, if you just look at that as a purely genetic experiment and not I mean, it is epigenetic, but if you look at just the genetic data in ENCODE that point to variants that are near enhancers and so forth. So enhancers are things that show tissue-specific function, and that's very much part of the epigenome program too. So a lot of the language sometimes get in the way of you know what I mean. It's like there's great science and all of these things that they can integrate really well. I think my goal in all of these years that I've been doing this, since really even I was doing cancer genetics, but certainly since we got our SEGS grant and I've been doing this recent work, is that you have to look at genetics and epigenetics as as integrated together. You can't think about epigenetics without genetics. I would argue that often you need epigenetics to understand purely genetic data too. That's what I think. Okay, so.
Well, what a wonderful presentation and a wonderful conversation here amongst all of you with our speaker. There will now be a reception in the NIH library where you can continue the conversation with Andy over coffee and cookies. Everybody is cordially invited to that. Come back next week. Meanwhile, let's thank our speaker one more time.
Thank you very much. Thank you so much.
Wrestling with how to pay for whole genome sulfite sequencing.